All right, thanks everybody. Thanks for coming today for the final meetup of the year. And we've got two really cool talks today. We've got Axayacatl Gonzalez from UQ, and we've also got Zacharie LeBlanc from QUT. So covering both campuses today, which is nice. And thank you to everyone who's joining us on Zoom. Let's start off by acknowledging the Turrbal and Yagara people as the traditional owners of this land where we're having where this is taking place. One of the best ways that we can acknowledge their elders is to recognize that there always has been teaching, research, and learning in this environment. And to everyone here and everyone on Zoom, if you're not already part of the SBA community, but you would like to take part more or find out what we're up to, you can get in touch via any of the avenues up on the screen there. And we've also started uploading recordings of these talks to YouTube. So if you want to check up on anyone's presentations or you missed a session, you can find the recordings there. All right, so yeah, today, first we're going to have Axayacatl Gonzalez. Axa has degrees in biotechnology and bioprocessing from the National Polytechnical Institute in Mexico. There he studied metabolic flux analysis in yeast and E. coli. And he came to Australia in 2013 to do a PhD here at UQ at the ARBN with Esteban Marcela and Lars Nielsen. And he completed his PhD in 2018. Since then, he's kind of become the hub of the systems and synthetic biology wheel over at the AIBN there, with postdocs in the ARC Training Center for Biopharmaceutical Innovation, and also the Queensland Strain Factory, a very exciting initiative that's been picking up steam in the last couple of years. He also collaborates with Amaris and the Queensland node of Proteomics and Metabolomics Australia. So he's your go-to guy for systems and synthetic biology. He's working to develop a high throughput strain characterization platform at UQ. Everyone, please welcome Axa.
Thanks. Hello, everyone. Thanks. I want to thank again Alex and Laura for organizing this part, and from the SBA initiative to kind of help us to be here together to share all the research we do in systems and synthetic biology. Okay, so that's kind of the title of my talk. So about the implementation of a high throughput phenomics, why, how, and what. Already James said a bit things about me. So again, I'm Axayacatl Gonzalez, full name Axayacatl, so short name Axa. I want to start to give a very short story about myself so you kind of get the context. I'm from a town in Mexico, close to Mexico City, called Xilotzingo, where it's which means where they praise the goddess of the corn, but also it's very famous because there's a lot of axolotls on the rivers. So those are kind of pictures of where my parents live and the churches. As James mentioned, in 2013, I moved to Brisbane, and was a huge change because Mexico City and where I live is almost 3K above sea level. And Australia and here in Brisbane were 28 meters above sea level. So the change was huge. And over all this time, I made a lot of growth as a person. And here I'm in a very strong position in terms of where I work and what acts I have to share my knowledge and share what I know and what I can learn. And in that case, I also want to mention that I'm also a member of the gay community. And I mention this thing just because it's something that matters. There's people out there that they don't feel represented, and just seeing us here, maybe for some people it's not important, for others it's very important, and for me it's important, so that's why I mentioned that. And the last thing, and you know what I mentioned this part, is one of the favorite books that I like ever is The Little Prince by Antoine de Saint-Exupéry. And I went to this book because kind of what I want to tell about the phenomics, kind of you can all go to this book and find a quote that actually fits there. And in one of the passages of the book, the little prince left his planet and goes traveling around and goes to this planet where there's one king that doesn't have any followers. But he claims that he can command everyone to do what he wants. At some point, the little prince is missing a sunset because his tongue was very small, so he can always watch the sunset all the times he wanted. And they say, and the king responded, okay, yeah, you will have your sunset, I shall command it. But according to my science of government, I shall wait until conditions are favorable. And how does that fit with phenomics and synthetic biology? We are at that point that we're able to manipulate biology to create almost whatever we want with some limitations, right? So we can command these organisms to build what we want. We want to produce alcohol, we can engineer it and we can do it. However, the only way that how we make that possible is making those conditions happen. And that's how all these parts of phenomics enters into the context of how we find the favorable conditions for whatever product we want to make happen. Okay, so then we go to phenomics, and then to understand phenomics, we need to go to the background of all this information, which is the genome to the phenome, right? Going back to high school, first year university genetics, they know, okay, the genome is the potential of an organism to whatever features they can express. The phenotype, the phenome, or the phenotype is how those instructions are expressed. And that kind of corresponds if you've ever been to a mirror house during a festival, right? You go there and then you go into the festival and you know you are this person, and then you look yourself through the mirrors and you see different versions of yourself. So you can see yourself there as you are this genome, and each version in the mirror is a different phenotype, right? So there's how that phenotype is different, how that each image is different is based on the conditions that the mirror allow you to put in there, right? So in that case, we have this traditional definition of what the phenotype is, which is the observable traits of an organism, right? Based on the hierarchy of the genome. It's kind of very basic, but now as science evolves, we have more accurate approach because now it's a set of observable metabolic traits to relate to the cells and the growth conditions, right? So now it's only like, okay, this is the genome and this is the phenome, right? For a genome, you have different phenotypes. And those phenotypes are not only based on how it grows, it's what the environment is, how it evolves, with what interacts, right? And in this term, we have a goal with the phenome, like to acquire a high dimensional phenotypic data, which means if we're able to capture the different phenotypes, and then we can track it down towards the genome of the organism, we can create the models, and then we can then predict from the genome what are the favorable conditions to make the phenotype to appear in a organism. If we see that in a context of spaces, we have a genetic space here, right? That is Q1 on the left side that expresses phenotype Q1. Probably just mention, put it here. Let's put it in this direction, right? So all from here to here, there are a lot of changes, right? Which media, the temperature, environmental conditions, media. But then this phenotype changes because once you put your cells in a culture, it doesn't behave the same way over the time. It changes. So the phenotype that you observe initially, which is lag phase, is different to exponential and to stationary. So that's the change to phenotype 1. If you collect that cell from there, the changes in reproduction create a new genotype. There were modifications in the environment to go to G1, and then they rearranged to a second stage of genome, right? So we have epigenetics, we have natural selections, but also depending of this, we can have changes in the lineage of how they reproduce, and then random mutations. So all these traits can be tracked. Then how new is this area of knowledge? Not relatively new. In 2001, Biologics started this platform, which is a phenotype microarrays, where they have all these micro plates where they could characterize the phenotype of a organism based on carbon condensation, nitrogen sources, chemical densities, pH. How they did it is based on a redox process in which they subject the cell to run different conditions. And only when the growth was favorable or like optimal, there will be a redox, the reduction of TB, which is colorless to purple, based on the activity in the mitochondria, and that is positive, it means that the bacteria show a positive behavior. So you build a phenomic landscape there. So there was a single layer integration, right? So it was like, okay, we have the genome and we want to identify if it grows on glucose and associate to a gene. If it's positive, the gene is there. If it's negative, the gene is not there or it's not bad. So it was like 1-to-1. Now, with the emergence of omic technologies, we have a new context. We have lots of data. So we have lots of data that we need to find a way to put it together, starting from the strain selection, and now first we need to find out what is in there, right? So what we have in the plate. So we do next generation sequencing it's giving us lots of data. Once you get that one, we enter on all this space of the omics, genomics, transcriptomics, metabolomics, that give us very specific information about how the cell is behaving, like transcriptomics, like really what genes are on, proteomics in terms of transcription, metabolomics, concentrations, just like regulation or any other information in there. If we put all this information together in a smart way, we can get an integrated phenomic analysis, which means we will be able to take from the genome, collect all this information, and then put it into what is a new system or like a phenomic platform that is able to integrate the data, have a complete annotation of the results, have also statistical data. That is something important, right? We can have one observation and that only happened once and we don't know why. So we need to make that is statistical. We have causality. So we really cause like A causes B instead of A might be causing B. A might cause B and then create new hypotheses and we create new platforms, right? And all these parts, okay, we can do it, but then what's the reason beyond that one? Like we can manipulate. Is there any reason why we need to manipulate? And there's a reason. This reason is associated to what is called this driving force. And what is called the second, like a green revolution, right? Initially, we have civilization where agriculture flourished, and we grow as a society. We created, and then we found out this development of these fossil fuel technologies. We exploit that. We are running out of that of those resources, but also we know we're going back to bio-based production from biomass that we can get the same products, right? So we're in this transition, and in this transition, we just in 1920 when we started developing all these new processes in a smart way. We have fermentation technology before, we have a lot of knowledge, but in a very rational way, we try to implement cause and consequence. It was is quite recent, and now we need to find better ways to do it, and that's why phenomics is important. Okay. Given the context, how we can actually put this to improve process, right? So how can we implement systems biology by process, taking into account what is phenomics? Okay, the first, and here's where we did how we do that. So which are the tools that we have here? And in terms of the group where I participate at UQ, which is the test lab, which is part of the Queen Industry Factory. In this case, it's okay, how can we go from these 96 plates that is giving us like this small phenome landscape towards a reactor, in this case, is just 500 meals, but can we higher scales? Well, for that, we have different tools, incubators, we have, and for that, we need to do high throughput, right? So we cannot only go and say, okay, we're doing one flask at a time. We need to increase the number of observations. That way, we can have a statistical information that is significant. So we have incubators, shaking incubators, plate breeders, and simultaneous force systems and multi-force A system of reactors and a gas analyzer that allows us to capture what is being produced by the system, by the reactors when they by the cells. But that's just how we observe the phenotype. Now we need to, okay, how we capture all the information, how we process the data. So for that, we start from tissue monetizing. So we need to recover what is inside the cell in a reproducible way, centrifuge to recover in phases, and now liquid transfer to robots to automate the process. We remove the human error, and we just we press a button and we let the robot do everything. So we can rely on the data we have were generated, right? Right now, what is the capacity that we have in this Queensland Straight factory? So right now we have, in terms of fermentation, high throughput fermentation, we have 8 simultaneous reactors, 200 to half a liter, 200 ml to half a liter batch of fed batch. We have online monitoring system, optical density, pH, dissolved oxygen, and we can analyze for each channel up to 32 channels on the mass spec, which is lots of data to process. And then we process all those samples, and we can do up to 96 simultaneous samples. Usually 24, because it's only me at the moment. But with the robots now, we can do the 96. And then we have this pipeline in the concentrate factory where we can kind of screen this phenotype from propagation, fermentation, and sample preparation in terms of protein sample, supernatant for certain metabolites, and cell type, which is like extraction, transference, and analysis. All this is done in the lab, so we can see this high throughput screening, have the reactors. We have to multiply our cells to be able to do all this job. And we have characterization, and we can observe different conditions. One of the things that we also do is characterize the fermentation and build strategies, right? This is one strategy we started with one system where we have a batch stage, then 2 fed batch with a harvest, and then sample. So we selected those points where we select and we analyze the data. And based on those implementation and doing some changes in the strategy, that could be either temperature, could be how much we feed in terms of a continuous feed or process, we were able to go in terms of this product that was originally a production yield in arbitrary units from 30, from A, up to 150, like 5 times increase, used by these changes, and with kind of statistical difference. We optimized that, and now we only have shorter fermentations that give us the same amount of data. But once we get all that data, how we process that, and that's, I'm the hands-on in the lab, but I'm not the only person to make this possible. There's a huge team. Well, we're still a small team that participates in this part, and the next part is my collaborator, Dr. Tim McCullum, that is not here, but he does all the analysis of this data and the rational design for the improvement of strengths. And he starts with this like data and the rational metabolic design. Based on what is in the data, he creates models. Let's start with this pathway that is only 6 reactions. Looks pretty simple, right? However, those simple pathways could be very complex. Let's start with this like drain towards biomass, right? So we usually see a set of reactions, A go to B, but also A and B are part of the biomass. So we need to take that into account in those analyses. Some of the enzymes within those pathways, and this in this process that we're involved, are highly complex, like P450s or PKSs, polypeptide synthesis. So that means that the system is more complicated. Some of the metabolites and enzymes are high promiscuous, means like not only A make A go to B, they also A to B to C to X or Y. And some of the compounds are toxic for the cell. When you try to engineer something, you realize that you're also killing your cell. And then also the system tries to regulate itself, forming metabolomes, that is groups of like enzymes that participate in pathways that try to keep homeostasis in the system. And also there's allosteric regulation. Whatever organism, whatever metabolite causes effect in the enzymes. So all these should be taken into account. And the way to do this part is using these metabolic models, right? In terms of what we call flux, that is like, we need to evaluate how much is produced of the flux is carried by an enzyme, and that flux is proportional to a driving force and the capacity available by itself, which the driving force is what is the thermodynamics that rule the chemical equations, and the capacity is the abundance of the enzymes within the cells. Once you get that one, we now go to the realization that the increasing flux doesn't mean that we increase in pathway expression. With like having more enzymes doesn't mean a higher flux or high productivity. In that case, we need to tune in the concentration of proteins in terms of feedback or feed-forward regulation, maintain those toxic metabolites in a low concentration, keep the balance. And in this case, we try to measure all this. Our goal is to optimize the flux, in that case of the final product we want to produce, based on those metabolomics and proteomics analysis that and from the samples that we get. And then we learn, and now we build kinetic models. That is much more like complex mathematical part, but that's how we address this problem of solving and design the overexpression targets. So once we know that, we can say, okay, that's what we need to overexpress, that's what we need to remove from the genome to make the phenotype that we want to exist. In this case, so we can go from a single strain and evaluate the fluxes, and then we need to sample. We need to make the model run several times so we can get a behavior, and then we can get a consensus of all the data generated. So we can generate multiple strains based on those hypotheses that we observed before and do the same, right? And just what is going to change is the number of observations we need to make so we can get statistical significance there. And eventually the cycle goes on and on. In this term, the current project, we are working with Ameris, where we are building two compounds from the aromatic pathway. They build the strains, we analyze, we process that, we generate all these, we apply these mathematical models to get the systems. And we send the science back and they build it. And we are now the third round in that part. So that's kind of in terms of how we put that together. But then in this case is a little part of what we do. We have other projects I will try to cover in the next 10 minutes before I finish this part. What also we do in terms of the phenome, right? So one of these is a PACO that we know this a strain from Propionibacteria that is acetate kinase knockout. So how this one started, so Propionibacteria is a microorganism usually used in the formulation of cheese that can produce propionic acid. And then there's many pathways to produce propionic acid. Our goal was to make a higher producer. The initial goal, okay, it also produces acetate, let's knock out acetate. But for knocking acetate, we need to build a molecular biology tools to engineer the system. So first we did some CRISPR knockouts with the help of Laura. She started that work with us, where we did a target knockout of histidine. We did remove histidine from knockouts. As you can see, only when you supplement histidine can grow, without histidine doesn't grow. We proved concept of that system. And then we said, okay, let's knock out acetate. In that case, there's no acetate production. However, what we found here in terms of the Cas9, when we remove this gene and we excise it from GFP, we kill the cells, right? Wild type and the mutant, wild type mutant, the cells die. We subject that to adaptive evolution and they survive ale. And if we just go to this, the last row here, there was improvement in the ratio of propionic acid to acetate, but they still produce acetate. So we knock out acetate, we kill them, they evolve, and they still produce acetate. And it was okay, that's kind of lame for us because that was not what we were expecting. We did all this analysis in the reactors. We have differences between the wild type strains and the mutants. And also the wild type doesn't produce as much carbon dioxide as the mutants. Interesting observation. But then we found this, the bacteria, when we did the ale, actually produces propanol and ethanol. We knocked out acetate and we produce ethanol and propanol that was never reported in that bacteria to be produced. So it was kind of like a interesting result. We demonstrate that with HPLC and also through mass spec, we calculate the MS by the ion. Here 45 is ethanol ion. It's only present if there's ethanol. Wild type doesn't have it, the mutant has it. What happened here, it's we activated SOS pathway, rescue pathway that goes outside of what it was reported from the Goodwardman cycle acrylate and the lactaldehyde pathways. So where we have propanol towards propanol, 1,2-propandiol towards propanol using this PDU operand that was silent and was activated. We're still working on all the details beyond, but that's something interesting that we found only when we did all these characterizations that I was not expected or we couldn't find in any other option. What other projects we're working on? This is another rescue, and this SOS3, that is a plant growth promoting bacteria. We started working last year. That what they do, they help plants to grow, right? There's different advantages of those bacteria, production of cyanophores, improvement of growth, production of hormones, right? What was important here is that when this bacteria was characterized, it was taken at different points during the growth stage, and every time that they took that bacteria from either early exponential or late exponential, there were different conditions, and we didn't know why, right? Like what was causing the effect in the plant. So first we started characterizing the plant. So sorry, the bacteria. We went from very low ODs, OD2 in a batch and pH, uncontrolled pH, to in a fed batch fermentations up to 15 or 30 ODs in fed batch. And we then analyze the different conditions across the stages. And in this case, it's worked on by one student, Alex. In this case, we evaluated different potential plant growth promoting effects. One of these is phosphate solubilization or indole acetic acid. So we can see in green that in most cases, in different conditions, we have production of those compounds, which is interesting because we now know that the bacteria can produce that while growing in a reactor. So we don't need to put them with the plant. So if we can exploit that and put that into further development, we can actually just produce the specific molecules that the plant requires instead of just let it grow with a microbial consortium. Then the final project that is also important for this high throughput phenomics is going from these genetic circuits we try to do to electric circuits, right? The basic principle is we want to build a sensor to identify growth, which means we know how a spectrophotometer works. There's light, there's a receptor, and there's an emission, and it's super expensive. We try to make a high throughput system where we can exploit all these very complex processes in terms of light and make something affordable. We're not the only ones. It's like those ideas are not new, but it's more like how we implement them, right? So one of the ones we started here is like using a Fitbit to detect the light intensity and then see the growth. The problem with this is kind of expensive and it's hard to get Fitbits here into Australia and more after the pandemics. So we went to much more basic systems with based on Arduino, which are like circuits that help us to control. So the principle is just if we emit a light here and we have a receptor here, the light is it detects how much light is here. If cells grow, the more light we have here, we have the less intensity, and we can calculate growth rates, right? What is the difference here? And that's important because we want to screen lots of strains. The challenges here is the design, connectivity, and reproducibility. So we need to make sure the design is good, how we put a lot of sensors together, and how whether it's important or not. And it's quite interesting because right now another student has developed a affordable biosensor that is less than $70, and that actually you can see the raw data here. So as you can see, the cells grow, the intensity goes lower, but the translation into OD is quite neat, and you can actually growth rate. It's this black box, that's the treatment bottle. $70 compare for a small bottle, and you can put that. Now we have 3. We are expecting to have up to 60 at the same time. And we can screen all of them, calculate growth rates, and well, we can even get some much elaborated system like alarms when it's in the maximum in exponential, take a sample. That's the next step, the high throughput. So as you can see, all those small projects come from all these ideas in terms of this phenomic landscape, how we can describe the cells. And this is only possible with all these people that participate in the lab and that support each other. We have, I want to thank to Dr. Zacharie, Professor Lars Nielsen, and also the metabolism team. They do all the sample processing, so they give us all this data we can work with HPLC and CMS. Postdocs, Camila Vero, that has helped me a lot. Laura at some point was there with us. Now it's the QT, collaborators, Dr. Chen, and the students that also I think are the core of the research that we do at any university. And just to finish here, it's another one to finish with another quote from Little Prince that says, "A rock pile ceases to be a rock pile the moment a single man contemplates it, bearing within him the image of a cathedral." We can now see those cells just as whatever country is there, but we can see them as the potential that we can build. So it's as long as we have in mind something new, something innovative, we now can do almost anything. Thank you so much.
Thanks very much, Axa. Any questions from the audience to start off? And if you've come and joined us partway through, please, you can come up to the main seat here. Yes, one on the front here.
with your Queensland, Australia, how many different strains have you put through by different species / what capacity do you have at different species, like bacteria species, archaea, things like that?
Thank you. That's for the question. In that case, we have in terms of capacity and the conditions, we can work with bacteria and yeast. We have grown E. coli in those reactors. We have grown this plant growth bacteria in the reactors, yeast, and depending on the needs, we can adapt for that. We can be anaerobic, aerobic, semi-aerobic. So it's quite versatile. And the strain also has been focused right now towards this AMREX project. Now we start to open that for other industrial plants that are interested in those analyses. Different bacteria, lactic acid bacteria, Pseudomonas, so almost any organism can be grown in those reactors based on the conditions. In terms of how many cells, so I think in the last 2 years, we have grown probably between 500 to 600 fermentations that last for 1 week in 8. The most we were able to process about 48 to 60 fermentations experiments per month when we're like the most easy. But yeah, it's quite versatile and maybe not so good for mammalian at the moment because of the impellers, but it can be adapted.
Yeah, so can I ask about the SOS3 bacteria? So I have 2 questions. Is it an endophyte? So is it living inside the plant?
That bacteria is kind of a mystery. It's an endophyte, yes, has been reported to be an endophyte, but also it's not spores and has different behaviors. So not only for one plant, we have different plants, and depending on the plant and the conditions interact differently. So the goal for that bacteria is, because it's so complicated to analyze interaction in the plant, we want to create this phenotype landscape so we can exploit what is in there and recover those products from the cell and put it into the plant. So that's kind of the approach we try to do with that bacteria. But yeah, it's an endophyte.
Okay, yeah, so the other question is basically that, but kind of the flip side of it, because you mentioned that approach where you're taking molecules that are outputs of that bacteria and artificially giving them to the plant. Yeah, I was just wondering if there's any consideration to the approach of seeing what that bacteria needs and providing more of it so that you could promote growth of that bacteria if it is helping the plant.
Yes, so in that we are more part of the reactor part, and also we do work with the plants. That is much more complex. It's agriculture sometimes been disregarded, but really the research potential in agriculture is huge, and in terms of synthetic biology is one of those areas that is kind of emerging now. Yeah, definitely there should be options to see how the bacteria interact with the plant, but that's not really my expertise or what I really want to cover because now it's already too complicated with the bacteria. Thanks.
No further questions? All right, thanks again very much, Axay. Let's just see, good point by Alex. Are there any questions in the chat on Slack? This is your chance. Sorry, on Zoom. This is your chance. Okay, no questions on Zoom. Thanks again, Axay. All right, next up we've got Zach LeBlanc from QUT. Zach got his master's degree in Canada and has spent a lot of time working in industry in the plant biotechnology field, which is really a kind of an underrepresented quadrant of our synthetic biology community here. So it's good to hear some of the plant scientists. He's worked in some really cutting edge stuff with companies in Canada that were developing plant-based production systems for therapeutics for HIV and prophylactics for nerve agent exposure, I understand. So really exciting stuff. And now he's doing his PhD here at QUT, and he's working with a very special and kind of semi-famous globally Australian native plant. I'll hand you over now to Zach. Thanks.
Thanks a lot, James. Yeah, so today basically I wanted to talk a bit about my work with the plant Nicotiana benthamiana, and what we're doing essentially to optimize this so that it can be a really fully featured biologics production platform. So the idea of this is essentially biofarming, and to simply put biofarming, it's taking an organism and genetically modifying it to produce a drug or a protein of interest, which you can then recover, which I mean is essentially what Axel was talking about as well, right? But this is a system that can be done in many different organisms. Essentially, all you do is you take your the sequence of your protein of interest and you pop that into your organism. Organism can be bacteria or yeast, insects, mammals, or even plants. And you take that organism and then you extract your molecule of interest. And in a lot of cases, it'll be a drug because it's an extremely high value product. So I'd like to focus on plants for this application. And the main reason why I believe in plants as a system for this application is that they have extremely efficient and economical production of biomass. So if you think about a plant compared to many other organisms, what are the inputs, right? You've got sunlight, dirt, and water. That's pretty simple to take care of and extremely cheap to grow. And the more biomass you have, that translates to more of your product of interest. So the plant that I'd like to focus on, and it's used around the world for this application, is Nicotiana benthamiana. This plant is an Australian native, and a reason that we use it for this application is that it's very easily transformed. And by that I mean it's very easily genetically modified. So I'm going to walk through a bit essentially what a standard process of biologics production in this system looks like from A to your final product. So you start by growing up your plants. You take seed that is genetically modified to offer some attributes that I'm going to describe later. But all you do is plant these seeds out. Isn't that nice? 5 to 6 weeks of growth. Boom. And then you take this plant and you bring it to a vacuum infiltration setup. So what we have here is a vacuum chamber, a vacuum pump, and a flask which contains bacteria. That bacteria is called Agrobacterium tumefaciens. So Agrobacterium tumefaciens is a natural pathogen for plants, and what it does is it transfers DNA into plant cells. Now, in lab, we've disarmed this pathogen and we're using it for our own uses by introducing our own sequences of interest. So an example of this, if we were trying to create an antibody, we take the sequence of our antibody of interest from number 1. We take that DNA coding sequence, we pop that onto a plasmid, we take that plasmid, and then we pop it into our Agrobacterium tumefaciens. Then we grow up a whole heap of it and use it in our vacuum infiltration setup. So proceeding with this, we dump that into our vacuum infiltrator unit. All right, take our plant, dunk it in, put on the lid, and then we apply the vacuum. So what's happening when we're applying the vacuum is that there's air between cells inside a plant's leaf. Applying the vacuum sucks the air out, and then when we release the vacuum, these spaces get filled by our Agrobacterium tumefaciens solution. So the bacteria is being directly introduced adjacent to all the cells in the plant's leaves. So then we're going to wait for about 7 days for our protein of interest to accumulate. While this is happening, our agrobacterium from the previous slide is taking that sequence that we put into it, conjugating to the cell with the pilus, and it's shooting that sequence into there. Our plant cell right here is producing our product of interest. Now for an antibody, we want this protein to be fully processed. So we want like glycosylation, correct folding, and all those things. So we would direct it to be secreted to the apoplast, which is this outside space of the cell. Following this, we'll take our plant and we'd homogenize the leaf tissue. I'm going to start by putting some buffer in our blender. Grab that plant, shake off the leaves, put on the lid for safety, right? And now we blend it up. And now we have our plant homogenate, right? So essentially what we have is a green goo that's leaves that are all mashed up into a buffer, and it contains our product of interest. Now that's not useful to anyone, but we would have to extract that drug that can be then used in humans. So to do this, we take our homogenate, which starts in this square here, and then we pass it through a first crude filtration step. Now this step is getting rid of all the large chunks in solution, and what's passing through are our antibodies, right? Following this, we'll do a tangential flow filtration step, and in here, our solution is passing through the center, and the small bits are passing out the sides through this tangential flow, and we're getting rid of it. So we're really selecting for the size of our molecule of interest, which is this antibody. Now in the next step, we're selectively choosing our antibody using affinity chromatography. So a lot of you might be familiar with protein A chromatography. So our antibody is specifically binding to these beads, and all the rest of the garbage that's in our solution is just washing through. And then we can specifically elute our antibody. Following that, we can polish it with a polishing column using a media like CHT, which again gives us very high resolution, and we'll specifically elute the form of our antibody that we want, even though at this point our antibody is about 90% pure. But after that step, we essentially have a final purified product that is likely of sufficient quality to go into a human. And that's the step from start to finish for biologics production in plants. Now, the part I didn't mention is that there's some issues with expressing biologics for human use in plants. And one of those is that plants and mammals make proteins differently. And the way they do this is through glycosylation. Glycosylation is the addition of sugars onto a protein. And on the right, I've given a diagram of a typical plant end glycosylation versus a typical mammalian end glycosylation. And there's some very clear differences here. Particularly, I'd like to point to the Xylose and the fucose groups that the plants have that are not present in a mammalian glycosylation. So this is a big challenge that you have to overcome if you're intending to produce a protein in a plant to go into a human, because if you put a protein in a human and it's got a glycan looking like this attached to it, it's going to be recognized as non-self, and it's likely going to cause the human to mount an immune response towards that glycan, that protein that looks like it came from a plant, right? So we've got to humanize the glycosylation profiles of our plants so that it's able to produce human-looking proteins, essentially. And to do that, we have to knock out the enzymes responsible for adding on this xylose and adding on this fucose. Now, the issue with that is that in the plant that I'm interested in, Nicotiana benthamiana, there are 5 fucosyltransferase genes and there are 2 xylosyltransferase genes that we'd have to knock out. And here's that family. So these are fucosyltransferase genes in benthamiana. You can see this cartoon shows
5 of them there, and these are the Xylosyltransferase genes. The cartoon shows 2 of them. Now I developed an experiment where I would actually knock them out using CRISPR-Cas9, a CRISPR-Cas9 system, and I used a polycistronic guide RNA, which allows me to incorporate 3 targets. So these 3 targets will go and hit these genes in these different spots, indicated by the colored triangles, and hopefully it'll cause inactivity to that gene. So essentially, I'm going to be knocking out activity of those 5 fucose genes, and with a second construct, I'd like to knock out activity of the 2 Xylose genes, which would eliminate these plant-specific glycosylations. And what I found after doing this is that, interestingly, I got different efficiencies for knocking down each of these proteins out of 10 lines that I analyzed. So in the first cassette, the green triangles, I got about 50% efficiency, and that really changed with the red triangles, which gave me 88% efficiency. And this is for making cuts in my DNA, which I verified in these plant lines by PCR. So what's next is to verify the actual protein that the plant's making. So I've gone and I've knocked out the transferases that are responsible for putting on these plant-specific glycosylations, but I mean, I have to prove that it's actually doing what I think it's going to do. So I have to check and make sure that this plant-specific glycosylation, this triangle right here, is gone. So I have a test that's an ELISA-based test that checks specifically for that sugar on plant proteins. And circled in, or I've got a box in red there around what a wild type plant looks like in terms of, in this assay, in terms of the amount of fucose that's on it. And all of these lines at the bottom are different lines that I recovered that have what appears to be extremely low signal of fucosylation, which I'd say is pretty darn successful for elimination of this plant-specific glycan. Now, the next step that I mentioned was taking off that star, which is Xylose. That's another plant-specific glycan. So using that second construct and using that line, P16 from this first diagram, I took that and then I introduced the second construct to remove that star, that other plant-specific glycan. And here we go in the second analysis. We can see that this P16, the second analysis actually checks for both of these glycans and not just the triangle. And this P16 line that we showed before had none of that triangle. Here, it has really high signal because it still has the Xylose group attached to it, right? But after I've introduced the knockout construct to eliminate xylosylation, I get a line like this, which is essentially devoid of all plant-specific glycosylations, which is fantastic. Like, this is the end point of the project, and I don't know how long you guys have been in science, but that's really rare to reach something like a definite end to a project. So I was pretty overjoyed to receive this line, or to recover this line. Now, part of this project, because I was concerned about knocking out 7 genes at the same time, and I heard about the inefficiency of CRISPR in plants. So I figured I probably have to, throw everything I have at it. And a really efficient way to knock out genes is through protoplasts. And a protoplast is actually a plant cell that's had its cell wall removed. So when you remove that rigid cell wall from a plant cell, you get these really nice spheres. And these are actually plant cells that are still living, and those green bits are chloroplasts within the cells. They've just had their cell wall removed, so they take on that most efficient shape, which is the sphere. And what these allow us to do is directly import things like DNA and proteins into the cell. And I figured this would be a much more efficient way to do this process, and it'll really allow me to get my end goal, which I showed you I achieved. I mean, spoiler alert, in the last slide, I achieved this. I was able to recover this plant without those plant-specific glycosylations. But I figured this is a method that I really need to use in order to get that.
because it has all these advantages, right? It's got higher transformation rates, higher mutation rates with CRISPR. And the protocol for doing this is you essentially take a plant leaf and you cut it up into little bits, you digest it with an enzyme, and then, as you can see, all the visible tissue is gone at this point, and what you're left with is these protoplasts, and they're really easy to transform. A big problem with this, though, is that there aren't functional protocols in the literature for actually taking a plant cell from this state to an actual plant again. So I think that's a big issue when starting a project, when the technology you're dependent on isn't fully developed. And I've been told before that it's good to include a joke in talks. So this is the timeline that I devised in the first year of my PhD as to how long it would take me to get this knockout line. I mean, that's the joke. Because I figured, developing this protoplast regeneration protocol, that's not going to be a big deal, right? I'll get that done. I think it said 3 months. That didn't work. I switched to plan B, which was traditional transformation, to get this knockout line, and eventually that worked. Glad I did it. However, in terms of the protoplast regeneration protocol, I just recently got that to work at the end of my PhD. So thankfully, I didn't rely on that to develop this line. But I think it'll be interesting to talk about nonetheless, because protoplasts are extremely difficult to work with. In this image here, I'm showing a protoplast exploding because it's in a solution that doesn't have enough solutes in it. So the cell's essentially taking on so much water that it's causing it to explode in this really dramatic way here. Bam. So there's all these features that you have to optimize if you're going to be working with protoplasts. And just from starting to isolate them, you have to optimize all these different components, starting with the osmotic pressure, which is the issue in this slide, followed by the agitation when you're prepping it, centrifugation speeds, and the digestive enzymes. All of these things can easily break this really fragile cell that's had its cell wall removed. So eventually I got to a state where I was able to isolate them and then culture them. And as you can see, I'm getting dividing protoplasts, which is fantastic, right? And I got to this point where I developed a protocol for regeneration of these protoplasts, right? From 0, where I isolate my protoplasts, encapsulate them in agar gel just to protect them and provide a solid medium for regeneration. And then all the while, I'm switching the buffer around these gel beads. So I'm taking out the old buffer, applying the buffer, and this allows my protoplasts to grow up, develop these small colonies, which get a bit bigger, which eventually I can go on a plate into these little colonies, which is fantastic. And you might be noticing the obviously missing part, which is shoot regeneration, right? Because that's not a plant. It's just callus. And that's a spot in this protocol that I got stuck on for about a year. I got them to this point, but I wasn't getting any plants back. I would just get division of this undifferentiated tissue. And there were some really neat things about this. Generating this stuff, I noticed, hey, this is cool. I'm getting these really big cells. And these cells were getting so big that in culture, I can look at them and I can see them with the naked eye. This cell is 0.5 millimeter big. Like, that's visible with the naked eye. And if you take a weak microscope, 20 times, you can observe cytoplasmic streaming in this cell, which is really cool. And they form these beautiful structures of undifferentiated cells with no chloroplast in them or anything. They just be this white growth, which is absolutely useless for regenerating the plant. So as fun as all this was, it wasn't useful. Where I made my breakthrough is when I took these undifferentiated cells and I played around with different hormone concentrations. And after trying, I think it was in the area of 90 to 100 different hormone concentrations, I was eventually able to get shoots regenerating, which is essentially the last part of this protocol, right? So this is regeneration of full plants from protoplasts, which I can tell you right now, because I've looked, there isn't a working protocol out there in the literature. Now, the last bit that I'd like to talk about is another component of this glycosylation part that I spoke about earlier. I mentioned that plants have these specific glycosylations that you have to remove, right? But I didn't talk about the mammalian specific glycosylations that you have to add on. And what I'd like to focus in on is galactose. As you can see, there's those two terminal circles on mammalian glycosylation, as well as the terminal bit is the sialic acid, which is that diamond. But that sialic acid is important for drugs because it affects properties of an antibody and how your body processes that antibody. So it's important to have those mammalian specific glycosylations added on to your plant produced proteins. And an absolute prerequisite to get the double sialic acids on the end there is to first have your galactoses added. Now, the way to do this is pretty simple. As I mentioned before in that workflow, we're adding our protein of interest into the agrobacterium. We pop that into the plant. You do the same thing, but in addition to your protein of interest, you're adding in an enzyme, a Xylosyltransferase, which will pop that galactose onto that glycan structure. There's a few challenges with this. One is that it appears to cause stress in the plant. The plant doesn't like high expression of this galactosyltransferase, based on things that we're seeing in the plant after introducing this galactosyltransferase. We're getting issues like low yields of our protein of interest. When we try to transform this stably, we get sterile lines, and we're also getting microheterogeneity, which is an issue, it's a quality issue with our protein of interest, because you see we want those double galactoses on the end. Microheterogeneity, we're just getting one, and that's not good enough. So one interesting thing that I found in the literature is that, yeah, I've tried to produce stable lines with this in the past, and they're sterile, which is no good, but they didn't have the same issue in tobacco. So what's that's telling me, because tobacco is a very closely related plant to this plant that I'm working with, is that there could be something specific about benthamiana that doesn't like this galactosyltransferase expression. As I mentioned before, this plant's used all around the world, but an advantage that we have here in Australia is that it's a native, right? So because it's a native to here, we have all these different genotypes that we can take advantage of. It's this amazing genetic resource that we have here in Australia that nobody else has. So the idea was to take this galactosyltransferase and try using it in not just the lab variety, the garden variety of our benthamiana plant, but all these different genotypes as well. Ecotypes, sorry. And what I did was I expressed my antibody without expressing the galactosyltransferase, and then I also expressed it with. And what I see in the light gray bars, you're seeing expression with the galactosyltransferase. The dark gray bars show expression without the galactosyltransferase. So you can see indeed in this standard variety, there is a reduction in expression that happens when you add this galactosyltransferase. So that's no good. But some of them, even though they have lower yields, they don't seem to be as effective. Like this ecotype number 1, it's the yields of your antibody don't change at all. Same with ecotype number 5, it actually seems to go up as well as ecotype number 3. So this is a really interesting result because it shows us that there's this genetic diversity out there that we can take advantage of that has properties that could be extremely useful for this process. We then further characterized these glycoforms using mass spectrometry, and this is with help from Lucia and Aga from University of Queensland. And the interesting thing here, I'd mentioned before, microheterogeneity was a really big deal to us. We've got one of these ecotypes, ecotype number 1, where we're seeing
heterogeneity. This is a really good feature, and this is something that we definitely want to take advantage of if we're producing this biologics pipeline in which we want consistent glycosylation, because it's giving us this final product that it's really desirable versus some of these other glycoforms, which, I mean, they have a lot of that microheterogeneity where you're seeing just that one galactose on the end. It's no good.
So just to summarize, what we've developed so far at QUT is basically an efficient, high-yielding system that can provide mammalian-like and glycosylations. And we've also got this amazing resource of these ecotypes from around Australia that we can take advantage of. And it really means that in Australia, we're well positioned to develop these types of systems for production. And these are systems that are deployed around the world, I guess mainly in North America at the moment, and some in Europe where they're producing drugs. A big milestone that happened in the past month was a company in Canada called Medicago actually just passed phase 3 trials for a vaccine producing plants. And it looks like it's going to be released for vaccinating against influenza soon. So that's really exciting because it'll be the first plant-produced vaccine out there on the market that's got approval for release in humans. Now, some future developments that I'd like to focus on are further optimizing the glycosylation pathways that I talked about and further analyzing these benthamiana ecotypes and seeing what other types of advantages they can bring to this plant biologics production system. And that's it. I'd like to thank ARC for funding this research, principal investigator at QUT that I'm working under, Peter Waterhouse, as well as contributions from Lucia Zaki and Naga at University of Queensland. And a special thanks to my colleagues at CAB, the Center for Agriculture and the Bioeconomy, located at QUT. And I'd be glad to take any questions. Thanks.
Thanks, Seth. And because you've given me the microphone, I'm going to give it first while everyone else can think whether they have a question. Having spent only a very small amount of time trying to implement Blast, the Protoplast work really blew me away. Were there any common rules in the end between the benth treatment and shoot regeneration with tobacco, or was it really like a clean slate trying to get the benthamiana to regenerate shoots?
No, yeah, so there were protocols for tobacco regeneration that I tried, and they didn't work, surprisingly. I figured, hey, this is close enough. I wonder why no one's ever published a protocol where they just take one of these tobacco regeneration protocols and repurpose it for benth. But unfortunately, it didn't work. There's a lot of similarities. However, I mean, there's a really great paper that put out a really detailed protocol for tobacco regeneration that works quite well. But unfortunately, it just doesn't work on benth. And there's a lot of differences, mainly being that it's an entirely liquid regeneration versus I have this sort of semi-liquid encapsulated phase where I've got these bubbles of protoplasts floating around in liquid media. So to answer your question, yeah, it's different enough to not work.
Okay, thank you. Any more questions at the top, Rob?
As a non-plant person, I work on what in that talk. So
Thanks.
Thank you very much for that. So the glycosylation in plants obviously has a functional role within the plant. When you knocked out the glycosylation that you did, was there any other phenotype effect?
No, that's the first question every time, and I should address it, but yeah, there is no phenotypes amazingly, and you'd think that we'd be more careful going into it and do an RNAi line, and in fact, that's what I did 7 years ago. Is I generated an RNAi line that did this exact same thing, and we went and patented it. It's great because it does this function, but
as you know, with an RNAi line, it doesn't quite have that definite phenotype where you're absolutely sure that the thing's knocked out. So, but yeah, no phenotypes in terms of the way the plant looks, grows, reproduces with the RNAi line or with this stable knockout, which is pretty amazing because it's got to have some important function, right?
So you've also got multiple chromosomes in the plant. Does that add a lot of complexity to the knockout strategy? And how stable is the stable line generation?
Okay, one at a time. So in terms of the multiple chromosomes, right, there's a lot of redundancy in benthamiana, and that's part of the reason why I had so many genes of that family that were so similar. So it did add a lot of complexity in terms of knocking them out, because I had to pick sites that were multifunctional and hit all of them, but also were specific enough to make sure that I got them all. So it's this weird balance, but then characterizing them through PCR, I've got all these genes extremely similar to each other that I've got to differentiate between. So for my sequencing reactions, I had to develop this nested PCR protocol that got more specific so that I could take advantage of tiny snip changes between these different genes in order to make sure that the edits were actually there. So yeah, it definitely added a lot of complexity. And the second part was,
yeah, just about stability and how stable the cell line is.
yeah, well, I'm assuming that it's going to be a knockout with transgeneration integrity, right? Because it's a knockout, and as far as I understand, it's not going to repair itself. But at the moment, I've only gone through 2 generations. So far, the phenotype's holding true. So it looks good. Thanks.
So really interesting you're talking about the plant cells. So I learned another word here. I'm just curious, when you talk about generation, you mean to get your plant to get a seed, or are you able to get seed that have genetically modified, or every time you have to do it again?
Yeah, no. So yeah, it's there's 2 parts to it, right? So you're starting the CRISPR knockout lines; those are stable, and that's essentially your host that you're transforming every time. So that would be your seed line that you're growing up, and that's not going to go away. So you have that stable line that you maintain, and then you're using that for your transformations because it's got the properties where it's removing those plant-specific ligands.
Amen. The protocol stuff was dope.
Thanks.
In regards to that, you mentioned you're using it because it's easier to insert information, easier to genetically move.
Based on the literature.
Basically, yeah. So now that you've developed this Protoplast protocol, do you think it does compare to your Argo bacterial protocol? Do you think it is a perhaps comparable or better way to go about gene-based works in plants?
Yeah, so it's hard to say because of my experience with it. I mean, if I were to start from fresh, I know how well the aggro works. Although to start from fresh, I wouldn't have to develop this again. It's a development that came too late for what I was trying to do with it. So at this point, I'm thinking, I'll just make a methods paper and maybe try it sometime. But in terms of the timeline and the transformation efficiency, I do see that it could have some advantages for sure. Yeah, I mean, to do a project like this again, I definitely give it a shot, but I don't have the data to say how it compares in terms of time. I just know doing it separately that it's a decent timeline and there's good efficiency, but I haven't put it head to head with the traditional agrobacterium stable transformation protocol. So I wish I could comment on it, but I can't really at this point.
And just with the antibody that the plant was producing, have you compared that? So I'm just trying to take out and improve the glycosylation of it. Have you compared that to the antibody that the mailing system would express? Like, have you done direct comparisons, such as that have that binding? What's the heterogeneity of the mailing antibody? Like, how does it perform in comparison to the mailing produced antibody? Or just without the scope for the project to actually compare that to the main antibody?
So it's outside of the scope, but I do have prior experience with that specific antibody. I was in a position to do research where I did that type of comparison, or I was able to see data from that type of comparison, and I can say that produced in this type of system, you can get an equivalent antibody that would pass tests to be a viable option to put on the market as a biosimilar. So a biosimilar being a drug that's equivalently similar to the innovator's product, that it can go on the market and take those regulatory shortcuts. So yeah, it's within the system you can produce antibodies that are just as good. And I'm actually doing some work with Lucia at EQ right now, where we're actually comparing an aglycosylated antibody produced in CHO cells and in BENT, and just see how they measure up. And in that one, we will be looking at functionality with a cell-based assay. So, stay tuned. I'll let you know.
Great talk. I always think about the commercial side of things. So, I guess, what are the advantages to producing an antibody in a plant-based system over a mammalian cell culture? I guess there's yield and those kinds of things. But can you comment on that side of things?
Yeah, totally. I mean, I've spent a good part of my career working on this system and working in this system. So I always have to ask myself, like, what's the value proposition? Am I wasting my time? And at the moment, the answer is no. I think this is a system that's had its growing pains in the past. A really good example is in 2005, when they were really excited about producing drugs in plants. So they popped a bunch of drugs into corn and threw them into a field. And then you had drug-producing plants going into other crops, and it was a complete disaster. The company went bankrupt. So, there's these early day examples of complete failures trying to use a system like this. But I think we've come a long way, and especially in the past 5 years, especially with the recent developments in the past month, where they're getting a vaccine producing plants through phase 3 clinical trials. I mean, that's viability, right? And now in terms of the advantages that this system has over other systems, again, I have to go back to the cheap production of biomass, right? I can't think of another way to produce biomass that efficiently, that cheaply. Now, the other part about this system that I didn't emphasize that I think is crucial is its flexibility. Because as you notice, we're growing up the plants 5 to 6 weeks, and then we infiltrate with agrobacterium, and then 7 days later, we're harvesting our product. So the determinant step in that for producing your drug is that 7 days, that 7-day period where you introduce the agrobacterium and the sequence of your drug, and then you harvest your drug of choice. Now that's a flexibility you're not going to see in many other systems because it's got that transient nature to it. I mean, a CHO cell system, you've got a factory producing, let's say, Herceptin, right? That's an entire factory that's dedicated to production of Herceptin in a CHO-based system. They're not going to switch it to another product in a week. It's impossible. A great example of this is Medicago, those guys that produced that vaccine. They got the sequence for the COVID-19 protein, and they were able to develop an antibody to it they were able to develop a vaccine to it in 19 days from receiving that sequence. They had a vaccine in hand, ready for testing, which is an amazing example of this demonstration of flexibility. And I think what's crucial to it is that with these other cell-based, fermenter-based systems, is that you're optimizing a cell for several things. You're optimizing it, and I think Axel can speak to this. You're optimizing it for growth. You're optimizing it for production. You're optimizing it to the conditions of the culture, right? These are all things, and that cell equals biomass. Now, in this system, the biomass and the production are uncoupled. You're producing a greenhouse full of plants. You produce your agrobacterium. You spray as many plants as you want drug, and then you get that amount of drug back. So this uncoupling, I think, is an extreme advantage because your biomass production isn't hindered by, like, your biomass production is separated from drug production in a way because you're just producing as many plants as you want. So I can keep going about it. I think that's a good spot to start.
All right, last chance if you're on the chat and you have a question for Zach. But it looks like we're all clear. All right, thanks again, Zach. And don't go anywhere just yet because we couldn't hold these events without our sponsors. And we've got a couple of representatives here today. We've got Peter from Decode Science. If you've been buying DNA in the last year, you've probably run into Decode Science already. Do you want to come up and say a few words?
Thanks, James. Thanks for having me. A couple of great talks here tonight. Exciting and yeah, very interesting, guys. Really appreciate being here. Nice to do something in person for a change. So I get to give away a voucher for a $500 twist voucher, and Alex has done some random draws for me, and the winner is Andres David. Thanks for your help, Alex. Andres David, who is at QUT. So Andres, if you're on Zoom or here, I'll send you an email anyway, and you tell me what you want and we'll make it happen. And also I had, let's go to the next email from Alex, a couple of twist prize packs to give away. So we have some socks and water bottle. So James Heffernan, if you're here, hey James. Aiden. Open the door. Oh, that was my best guess. Thanks again. Appreciate you guys having me here, and yeah, thanks for that. Cheers.
All right. Okay. Well. You've made some beautiful slides about the different organizations that you guys distribute for, but most of the audience is probably familiar. But we also want to thank NEB. Everyone loves the NEB, and Jenny is here today representing NEB. She's declined to give a spiel since we all know what they're about, but yeah, we really appreciate the support from both of you guys. Thanks very much. And with that, yeah, thanks again, everyone, for coming, everyone for tuning in online, and we hope to keep growing the Brisbane chapter of SBA in 2021. Yeah, stay tuned for announcements about the first meetup next year. Thanks.